Hi everyone, and welcome to this presentation on carbon fiber microelectrodes. Today we'll cover four main topics. First, we'll discuss the structure of carbon fiber microelectrodes. Next, we'll highlight the strengths of these tools. As with any technology, they come with their own set of challenges that we will explore, as well as ways to combat those issues. Finally, we'll look at the applications of carbon fiber microelectrodes. From medical diagnostics to environmental monitoring, these electrodes have a wide range of uses. Thank you for joining us, and let's get started. We will start with exploring the structure of carbon fiber microelectrodes, which are known for their incredibly small size and high precision. As shown on this slide, these electrodes have a typical diameter of just 5 to 30 micrometers, which is about 10 times smaller than the diameter of a human hair. A typical carbon fiber microelectrode consists of a central carbon core surrounded by a glass insulation layer with some of the carbon fiber exposed. The exposed carbon area is crucial for their functionality, and the length can vary, although we tend to cut it to around 100 micrometers in length. This exposed section, as noted by the picture on the right, allows for precise measurements and interactions at a microscopic scale when measuring in tissue. One great thing about these carbon fiber microelectrodes is that you can adjust their geometry based on your needs. If you are working in solution or in tissue, cylindrical electrodes are commonly used. However, if you bevel the carbon fiber down to the glass seal at an angle, you can create a disc carbon fiber microelectrode that is great for measuring exocytotic release at single cells. To form a recessed electrode, you can bevel the carbon fiber down further to create a crevice inside the glass insulation, which helps prevent diffusional loss of your analyte in single cell preparations. You can also modify cylindrical electrodes with enzymes to be able to detect non-electroactive species, which we will cover more thoroughly in our video on enzyme-modified electrodes. Lastly, you can also forego the glass insulation completely and use a silica-based insulation instead. This will create a flexible sensor that can be used for experiments in awake and behaving animals. Next, we will focus on the strengths of these tools, which make them an exceptional choice for numerous applications. First, they are biocompatible. This means they can be used within biological systems without causing adverse reactions, a crucial property for medical implants and sensors. Secondly, these microelectrodes are resistant to fouling. Fouling can degrade performance over time by blocking active sites on an electrode's surface. Resistance to this ensures long-term reliability. Although carbon is historically known to foul, certain techniques such as fast-scan cyclic voltammetry allow the user to sweep over a range of positive potentials. These positive potentials can essentially clean the carbon surface of your electrode. Next, these electrodes are inexpensive and simple to fabricate. In terms of performance, they offer good spatial and temporal resolution. This enables precise measurements over both small areas and short time intervals, essential for dynamic environments where changes occur rapidly. Their compact size allows them to fit in small spaces, making them ideal for intricate applications where space is at a premium but functionality cannot be compromised. Lastly, IR drop is reduced, which refers to the internal resistance of the electrode material. Carbon fiber microelectrodes are designed to minimize this IR drop, ensuring more accurate and reliable data. Now we'll focus on the challenges associated with carbon fiber microelectrodes. Interferences are a major struggle that can affect their performance, including but not limited to pH fluctuations, co-released electroactive species, and more. Managing these interferences is crucial to ensure accurate and reliable measurements. Next, carbon fiber microelectrodes are incredibly small and delicate, which can be a good thing, but also makes them prone to physical damage. This fragility requires careful handling and specialized equipment to maintain their integrity during use. Lastly, inconsistent fabrication is a significant challenge. Variations in the manufacturing process can lead to differences in electrode performance, which can affect the reproducibility of results. Ensuring consistent fabrication is essential for reliable and repeatable measurements. The great news is, though, that there are steps you can take to minimize the effects from these challenges. So what can we do to address these challenges? To address interferences, we can modify the electrode surface with a coating or something similar. By altering the surface properties, we can enhance the electrode's selectivity and sensitivity. Next, employing a reference electrode is absolutely crucial. A reference electrode helps maintain a stable background potential, allowing for more accurate measurements by compensating for any fluctuations in the environment. Out of any of these strategies discussed today, using a reference electrode is the most vital to ensure you receive quality measurements. Lastly, we can use background subtraction techniques such as Fasken cyclic voltammetry. This method helps to get rid of any potential interference that remain relatively constant in concentration, providing clearer and more reliable data. To mitigate mechanical fragility, we can reinforce the electrode with composite materials such as silica. We can also apply a surface coating such as Nafion for an additional layer of protection or use a protective casing. All of these steps can safeguard the microelectrodes from external impacts and handling damage, ensuring they remain functional in various conditions. To tackle inconsistent fabrication, we can standardize fabrication protocols to ensure that each microelectrode is produced with the same high-quality standards. We can use uniform materials to minimize differences in the properties of the microelectrodes. And we can also calibrate the finished sensors to correct any deviations and ensure that they meet the required specifications. Now that we've explored the properties of these electrochemical tools, I just want to highlight some of the diverse applications of carbon fiber microelectrodes. Carbon fiber microelectrodes are essential in neuroscience for recording brain activities. They help in detecting neurotransmitters and measuring neuronal action potentials, providing valuable insights into brain function and disorders. These microelectrodes are also used to measure electrical activity in biological systems, making them crucial for studying the electrophysiological properties of cells and tissues. This application is vital for understanding heart rhythms, muscle contractions, and other physiological processes. Another huge benefit of using microelectrodes is that they enable us to measure in non-aqueous solutions or solutions with high resistance, which expands the chemical repertoire of electrochemical monitoring, including things like biosensing and heavy metal detection. These electrodes are also highly sensitive and can detect minute changes in chemical compositions, which is essential for medical diagnostics and research. Lastly, these microelectrodes play a significant role in environmental monitoring. They are used to track environmental conditions, such as detecting pollutants and monitoring water quality, helping us maintain a healthy and safe environment. By leveraging the unique properties of carbon fiber microelectrodes, we can advance research and applications across these diverse fields, leading to better scientific understanding and improved quality of life. Thank you so much for watching.